Said I'm going to be talking about working with multiple samples in Giotto. So up until this point, we've kind of been having um, one Giotto object for one data set, but Giotto also allows us to group multiple objects into a single data set. And so this is important because that allows us to perform analyses on multiple data sets in conjunction with each other. And so we can use this for ensuring that normalization is consistent across datasets, and this allows us to also directly compare datasets. This can be done in any of the X, Y, or Z axis. So if you've got, say, like a 3D data set, you can group these in the Z axis to create a 3D projection of your data, or we can just do it in the X, Y space in order to compare data sets in that plane. Um, so this can be important for things such as like having multiple replicates or having similar tissue types. So whilst it's possible to integrate data sets, and this is relatively easy in Giotto, batch effects and differences between samples can also hinder integration. So in such cases, more sophisticated methods might be needed to successfully integrate data sets and to cluster them successfully. And so one such example is Harmony, and we're going to go into that a little while later. So for this tutorial, we will demonstrate the integration of two Visium datasets and examine the results before and after Harmony integration. So just a little bit of background on the dataset. So we're going to be using a 10x genomics dataset with an adenocarcinoma prostate sample and a normal prostate sample. So Jocelyn went over Visium technology in the day 1 and day 2, but just as a brief recap, we for this tissue was placed onto specialized slides, which contain cells or spots that have primers that can bind cDNA. So when we place the tissue on the spots, we're able to extract out the cDNA. These bind to the spots, and we're able to sequence and relate these back to spatial locations. So jumping over to the code side of things, we need to start by first of all downloading the data set. So if you haven't done that already, please do so. So we've got two data sets again, the adenocarcinoma and the normal prostate sample. So after you download those, if you could please just extract those using this code, and these should all go into the data directory for this session. So I'm going to kind of go over these initial steps a little bit quicker because Jocelyn covered them in great detail in day 1. But if you are having trouble, please just chime in and let me know. So the first thing we need to do is set up a Giotto object. And so as before, we need to set the instructions, if that wants to actually load Giotto. So we first need to set up instructions. So we've got our save directory, whether we want to save or show the plots, as well as our Python path. So for most people, that should just be set to null. But if you need to set that manually, please do so. From here, we need to load in the Visium data. So we're going to start by creating two individual data sets. The first is for the normal prostate. So we're going to call it npros. And so again, we have the Visium directory, the expression data that we want to use, the image that we will be using for this, as well as where the gene column is in our index, and the instructions that we defined above. And we'll do that also for the cancer prostate sample. So it's all the same, apart from we're going to change the Visium directory for that. So then, so from here we can take a look at the Giotto object, and we have cell data, raw RNA data, as well as a single image, as we would for most Visium data sets. We can also take a look at the tissue coverage, so we can see where we take a quick look at the layout of our data. So in black is where there is no tissue, and the red is where we have tissue. So we're seeing the overlap of Visium spots to the tissue. This is for the healthy data set, and then again for the prostate data set. So we can also take a look at the we can take a look at the makeup of our data using the p.data.dt function. So we can see all of our cell IDs, whether they are in tissue or out of tissue. So 0 if it's out of tissue, 1 if it's in tissue, and where the where the Visium spot lies on our sample. So at the moment, these are just single data sets. So if we want to join these in Giotto, we can use the join Giotto function, or join Giotto objects function. For this, we need to supply a few parameters. So we need to create a list of Giotto objects. So we've got our N prostate sample and our cancer prostate sample. We also need to specify Giotto object names. So these are going to be the names that are assigned to each data set. So we've got NP for normal prostate and CP for cancer prostate. So these can be anything that you want, but please, we need to make sure that the total number of data sets and the total number of object names is the same. We then have our joining methods. So for this, we're going to use shift. So this is going to just tell us to shift it in the XY plane, but we also have, if that would work. We also have other options such as ZStack if you had a 3D data set or just no change. We also have this variable here, X padding. So the way Giotto joins objects is it will take the images for each object and then space those out in the X plane. And so this X padding is just going to give us a little bit more distance between the data sets. But we can also tell the function to include Y padding if you want to pad in the Y direction as well. Cool. So we run this through. It's going to work. So we can now take a look at our Giotto objects and how it's changed. So we've now combined the data and made a new Giotto object called combined pros for the combined prostate sample. So we can see that nothing has really changed here. We still have cell and RNA, and that's expected because these are still the units that we're working with. But the main change so far has been these images. So we now have 2 images. And so Giotto will automatically update the image name to append the object name to the beginning of each image to create a unique image for each data set. And so we can also take a look at how the p.data output changes. So for each cell ID, we can see each cell has an appended object name at the beginning. So again, to make sure that we have unique cell IDs for every single data set. And we also now have this list ID, which is the name of our data set that we supplied. Cool. So now if we want to visualize the combined data sets, we're able to visualize them in the same plot. So if we plot this, we can now see that both data sets plotted within the same Giotto plot. And as I said before, with the X padding, we can see the space in between. And so that's what that was doing. It's just basically meaning that the images aren't going to be right up next to each other. So again, this is what we saw earlier with just in tissue, out of tissue, but within the same plot. We also might want to visualize data sets within different plots. Oh, sorry, what I should have also mentioned here is we now need to, if we're showing images, we now need to supply image names because we've got multiple images within the data set. So we need to tell it which images to visualize when visualizing with a combined data set. So we can also view these in multiple plots. So to do this, we're going to be using the group by function. So if you remember in the p data dt output, we had the list ID column, which defined our data sets. And so by grouping them by that, we're able to visualize the data sets in separate plots. And again, we still need to specify which image, so the NP image and the CP image, to tell it which images to render. And so if you want to, you can also split data sets up. So we don't need to keep them as a single data set. So to do this, we are again going to use the list ID. So if we get the p data output, so if I could drag that up, we can see the list ID for both the NP and the CP sample. And so say we wanted to make a new Giotto object with only our normal prostate, we can subset this for just our normal prostate sample, and then we can take the cell IDs from this in order to tell it which cells we want to subset. And so then we're going to use this subset Giotto function. So we're going to supply in the combined prostate as our Giotto object, and then we're going to tell it the cell IDs and the poly info to subset from. So that's going to be these cell IDs that we see below. And then we want to tell it which spat units we want to subset. So you can tell it all if you want, if you've got multiple spat units, or you can specify individual ones, such as we have cell. So we could just tell it cell, but both of these would work. And so now if we visualize NP split, we can see that we now only have a single data set. So from here, it's actually very much similar to what Jocelyn showed on Monday. So we're just going to run through the same analyses for that you would as if this was a single data set. So we're going to begin by removing any of the spots that are not covered by tissue. Then we're going to filter the Giotto object. So this is removing any of the features that are found in only a few cells, or removing cells that don't have many features within them. We're going to normalize the samples. And again, this would run. This is important because by normalizing these samples together, we can then directly compare the expression data between the different Giotto objects, which you would not be able to do if you were to normalize them separately and then join them at a later date. So we're going to add statistics, and we can then plot these and take a look. So we can automatically see there is actually differences between these samples. So in the cancer sample, we see a much higher number of features per cell compared to the normal prostate sample. And again, if we were to compare these separately, the way the normalization works, it would make it difficult to see these differences or to visualize these differences. So by joining, we're able to better see how different samples are to each other. So from here, we can then cluster. And again, this is all done in the same way as if you had a single data set. So we're going to begin by calculating the highly variable features, and some of these are going to take a while. So something to keep in mind is the more data sets you combine, the longer a lot of these steps are going to take, and they might require a lot more memory. So like with Visium data, it's quite straightforward, but if you were to get into some of the more in situ sort of spatial data, some of these steps might take quite a while to run. So bear that in mind when deciding on how you're going to approach these scripts. So again, we're creating a nearest neighbor and then performing Leiden clustering on our samples. So we can then run UMAP, and finally, once that runs, we can visualize this. Again, please, if anyone's having any issues, just chime in and we can help you. So now we've clustered both of these samples together, and we can see how they relate. So what we actually see here is kind of two separate groupings on the UMAP space. So this bottom left one here maps to our healthy prostate sample, and the top right one maps to our cancer prostate sample. So what this is kind of telling us is that these samples are clustering independently of each other. So even though they share like cells, they're not clustering together. And we can kind of confirm this by visualizing the clusters using the list ID to visualize the clusters. So remember, the list ID is telling us which sample is which. And so by telling it to color cells by list ID, we can see the contribution. So we see the top right, as I said, is the cancer prostate sample, and the bottom left is the normal prostate. And there's a little tiny bit of overlap between them, but for the most part, there is no overlap, indicating that if we wanted to cluster these cells together, we would need to use a more sophisticated method. So as I alluded to earlier, that method is harmony. Well, one of the methods we can use is harmony. So harmony works by trying to increase the diversity of clusters. So it's taking the PCA space and manipulating it in order to cluster cells from different samples together. So the way this works is it first of all takes a soft assigns cells to clusters. So it kind of favors mixed representation clusters. So in this case, representation means having different data sets within the cluster. And then it gets the centroids for each of the data sets. So that's for both so it creates centroids for both data sets independently, and then creates a data set correction factor for each cluster, and then uses this correction factor to move the cells based on their soft clustering membership. And it does this in a sort of iterative process until we reach a convergence where we have clusters that have a diverse makeup, so contain multiple different samples to better represent the to kind of correct for issues with clustering. And so again, we might want to use this if we have, say, batch effects from, like, say, running samples on different days, or if you've got control and treated samples that might not cluster together nicely. So it's also important to note that Harmony only manipulates the PCA space, and it doesn't alter any like expression data or anything like that. So if you're wanting to do any expression work, it's still possible with when running Harmony. So before running Harmony, we need to ensure that we have run PCA. If we haven't, Harmony has a do PCA variable. So if you haven't run PCA for whatever reason, we need to set that to true, but above we did run it. So for this, we'll use false. And there are a number of other variables that we can use, such as sigma, which determines the width of the k-means clustering. So larger values allow cells to belong to more than one cluster, and smaller values making the cluster approach more rigid. For theta, it's a parameter that controls diversity within clusters. So higher values leads to more diversity, lower values, less diversity. And lambda is just a parameter for penalty parameter for ridge regression that helps prevent overcorrection. And in clusters, which we will set for null for this, just determines the number of clusters that we want to have in our model. So we also, to run, what I should have said is to run Harmony in Giotto, we have the run Giotto Harmony function, which takes in the Harmony function from Harmony package and runs it in a Giotto specific manner. And we just need to supply the Giotto object as well as the variable to be used in order to define the diversity. So in this case, it's list ID, which is defining our samples. So if we run this, we're going to it's going to perform a number of iterative runs through Harmony, and will eventually converge. So if we ran this successfully, it should converge after about 5 iterations of the Harmony algorithm. And what I should have also said is, if you need more information on Harmony, the link is there, or we have links to it in the markdown. So please do read up on that if you're interested. So Harmony, as I said before, kind of manipulates the PCA space. So we're going to be using Harmony in lieu of the PCA going forward. So we're going to repeat the clustering steps, but this time by using Harmony as our dim reduction rather than PCA. And so we're going to actually be creating new sets of the same analyses that we performed above, but with Harmony. So if we run this now, we're going to be creating a nearest neighbor network that's called NN Harmony. And so we take a look now at our Giotto object. We can see that we have a dim reduction, we have Harmony, as well as nearest neighbor Harmony. And so we're able to keep all of these in the same Giotto object without overriding anything by specifying new names when we create new variations. And so going forward, we can then also perform Leiden clustering. And again, we're going to change the name to Leiden Harmony so that when we run it, it will appear alongside oh, it's not there. No, not there. But all of these will be stored within the Giotto object, and nothing will be overwritten. So then finally we'll run UMAP. And this will take a little while to run. So again, we're changing the name to UMAP Harmony, and then we can perform the same spat dim plot 2D to visualize the clustering. And so this time we can actually see again two different distinct groupings within our UMAP clusters. And so, but what we can see is that they actually map to both. This one on the left maps to both the normal and the normal prostate and the cancer prostate. So like, for instance, the blue here, here. This one on the right, though, only maps to the cancer prostate. And we can confirm all of this by, again, using the list ID to color our cells. Sorry, what I should have also mentioned is under like the dim reduction name for our splat plot, we've now changed these to UMAP harmony rather than just normal UMAP. So if we run this, we can see that there is mixed population here on the left, which is what we were expecting, and on the right, as I said, is just from the cancer prostate. So you might be thinking that this didn't work, but this is actually working as we would expect. So the sample on the right has cancer cells, whereas the sample on the left does not have any cancer cells. So what we're able to actually do is cluster together the like cells, so the normal healthy non-cancer prostate cells, but retain the cancer cells as their own unique clustering. So we're not performing over-clustering by integrating the cancer cells with normal healthy prostate samples, meaning that we're able to retain biological information whilst grouping things that are similar. So that's all we have for this section. So if anyone has any questions or if they had any problems, please let me know.